Hello everyone, and welcome to episode 52, Remastered. So far in this series on the physiology of plants, I've discussed how plants develop and grow, how they extract water from the soil, and how they absorb and process their nutrients. Today's episode will be all about another fundamental quality of plant existence: their senses. Plants don't have eyes or ears like us, but they do have a number of sophisticated internal chemical signaling systems and defensive responses. Plants don't have brains like us, or sentience, but they do have a unique kind of biochemical-based awareness of their bodies and the world around them. They can detect gravity, so they know which way to grow their roots and their shoots. They can detect sunlight, so they know where to best position their leaves and allocate resources for vegetative growth. They also have to have some kind of awareness of the time of day and the time of year, so that they know when to bloom, so that they know when to drop their leaves and go dormant for the winter, or when to pump up investment in growth for a coming growing season. While this kind of floral awareness isn't sentience or sapience as we understand it, plants are able to learn about their environment and make choices so as to maximally benefit their growth and survival in whatever environment they happen to find themselves in whatever environment they happened to germinate in. If this whole concept of plant sensoria still seems rather vague and unclear, just get comfortable and relax, because I'm going to explore pretty much all of this in greater detail. To describe these plant sensory systems as simply as possible, they have various types of specialized sensory cells, which can detect some kind of input. This sends an intracellular signal to other cells elsewhere in the plant's body, and those cells, on the receiving end of the signal, will make the appropriate response. If they get a signal that temperatures are dropping and the photoperiod is getting shorter and shorter, they might respond by preparing for winter. If they get a signal that gravity is this way, then the shoot will grow that way. Now, I'll start by talking about this sense of gravity, because I find this particularly interesting. The ability for plants to detect gravity is called the gravitropic response. Do you recall the structure on the end of the root called the root cap? I briefly talked about this in the last few episodes, but the root cap is basically a shield of dead cells on the apical side of the root meristems. The dead cells in the root cap protect the fragile stem cells in the meristem, as the dead cells are designed to be sloughed off against the rocks and the dirt that the root might bump into. But the root cap has another function. It possesses a group of cells in its center called amyloplasts. These amyloplast cells are partially filled with a handful of starch granules. These are dense little blobs, and they sink in the cell's cytoplasm. Gravity will pull them down, and they'll fall against some spot on the membrane of the amyloplast, on whatever point is at the bottom. Proteins in the membrane are able to sense where the granules are. They're able to sense the pressure. And then, through these proteins, the amyloplast becomes aware of where the starch granules are resting, and the amyloplast can tell the plant which direction gravity is pulling from. And this passing along of information encourages the root to grow downwards. But the question becomes, how does it encourage the root to grow downwards? How does moving this information work on a chemical level? In this circumstance, the signaling chemical is called indole acetic acid, or auxin, which is a powerful hormone that exists at extremely low concentrations, like a few billionths of a gram of auxin per gram of plant tissue. When the root is growing straight down, auxin is being actively transported through the plant's root tissue, down to the root tip and back up. This is a pretty stable cycle that sees the auxin distributed equally by proteins throughout the root's tissue. But when the root isn't growing vertically, sensory receptors will detect this and inform the proteins to move the auxin more towards the downward side. Auxin accumulates in the lower half of the root like water in a cylinder lying on the floor, and this increased concentration of auxin inhibits cellular growth on the underside of the root. This is really interesting because auxin is revealed to be a growth regulator. In steady, stable conditions, all of the cells will get a steady amount of auxin, and they'll all grow at a steady rate, straight down. But when some cells get dumped on with auxin, their growth is inhibited. Conversely, the cells in the top half of the root are exposed to less auxin, and so their growth is uninhibited. This creates asymmetric growth in the root, where the top side of the root tissue is growing faster than the tissue on the bottom side, and this causes the root to curl and point downwards again, re-establishing that vertical growth pattern. While gravity is a constant variable, unmoving and perpetually pulling downwards, other variables like light and temperature are very variable, both throughout the day and from season to season. Plants have a variety of responses to a variety of different wavelengths of light that they're exposed to. One of these responses is called phototropism, which is a basic response where the plant's body turns or bends to face a light source. Specifically, plants will only engage in phototropism when they're being stimulated by high-energy blue light. The blue light excites electrons in a variety of photoreceptor pigments, which all induce different responses. Some of these blue light photoreceptors regulate flower growth and the opening of stomata. Specific photoreceptors, called phototropins, are pigments that exist in the tip of the growing shoot structure, and these respond to blue light. By absorbing this light and getting an electron all excited into a higher energy state, the phototropin protein will undergo a conformational change that opens it up to phosphorylation. When ATP is used to phosphorylate the phototropin, the protein will undergo a second conformational change, and this will send a signal to auxin-transporting proteins, telling them to alter their distribution of auxin. Yes, this is the same chemical auxin that helps the roots bend downwards, but in shoots, auxin has the entirely opposite effect. In the roots, auxin gets pooled in the lower side of the root, and this high concentration of auxin will inhibit the growth of the cells. Meanwhile, the cells on the opposite side, on the upper half of the root, have their growth unhindered because of the relatively low auxin concentration, and so they grow larger, and the combined effect bends the root downward. But in shoots, the phototropins send signals to the auxin-transporting proteins to have them shuttle auxin only down the dark side of the shoot, down the side of the shoot that's not exposed to the blue light source. Because auxin here has the opposite effect as it does in the roots, this increased concentration of auxin in the cells on the dark side of the shoot will stimulate those cells to grow larger, while inhibiting the growth of the cells on the light side of the root. This will cause the plant stem to bend towards the light. But these cells in the stem that are doing the bending, they're already mature cells. They have hard, stiff cell walls. So how can they bend like this? What's going on? The answer is proton pumps. So the signal that gets sent out by the phototropins ends up stimulating the proton pumps, who begin fervently pumping out hydrogen ions, or protons, into the cell wall itself, which makes it a particularly acidic place. The low pH will activate enzymes called expansins, which then go and break a few of the hydrogen bonds holding the polymers in the cell wall together. This controlled degradation of the cell wall allows it to loosen up and become a bit more flexible. This is really important because the cell wall can't bend, but it can loosen to accommodate a membrane that's turgid with water. Consider the fact that the high proton concentration inside the cell wall also makes the outside of the cell positively charged, which makes the inside of the cell relatively negatively charged. This relative electronegativity ends up attracting positively charged ions to enter the cell, which will then raise the cell's internal solute concentration. As I discussed in great detail in episode 50 on plant hydration, a high solute concentration creates an osmotic gradient that will draw water into the cell. This incoming water will fill the membrane until it's taut and turgid and pressing up tightly against the inner surfaces of the cell wall. But now add the fact that auxin stimulation has made the expansins loosen up a portion of the cell wall. So now, when the membrane reaches turgidity, the internal pressure will push out against the loosened part of the wall, and the cell as a whole will expand in size. Thus, auxin activates the growth of these cells on the dark side of the shoot, and causes the shoot to bend towards the blue light. Photosynthesis is also stimulated by various wavelengths of red light, and these red wavelengths cause their own set of responses. One of the more interesting responses to red light is called the red far red switch. One of the wavelengths of light typically found in direct sunlight is a red wavelength around 660 nanometers long. Far red light includes wavelengths that are much closer to 735 nanometers, and this is the light that rarely gets absorbed, that instead filters through the leaves to hit the forest floor. Because this far red light isn't readily absorbed by the leaves, it's one of the only wavelengths of light that can pass through a lot of the canopy. If a leaf is getting nothing but this far red light, that tells the leaf that it's in the shade, that it's being blocked out by other leaves, perhaps on other plants, competing for the sunlight. Where red light stimulates growth, far red light suppresses growth. This idea of a red, far red switch was created as researchers were studying the enzymes and the pigments involved in absorbing all of these wavelengths of light, specifically of red and far red light. They called these photoreceptor pigments phytochromes, which actually absorb both red and far red light, but only in particular conformations. When the phytochrome pigment is in the P sub R conformation, it will absorb red 660 nanometer light, and this will change the protein shape into the P sub FR conformation. P sub R and P sub FR really just refers to phototropism sub R, so sub red light, or phototropism sub FR for sub far red light. These are just designations telling you which conformation the phytochrome pigment is currently in. When the pigment is in the P sub FR conformation, it will absorb far red 735 nanometer light, and this will change it back into the P sub R conformation, where it will absorb red light. In this way, exposure to red or far red light is like a switch that moves these phytochrome proteins between these conformations. So, what does this switch do? What does it turn on and off? Well, when the phytochromes are in the R conformation, which means they've been exposed to 735 nanometer light and they've changed their conformation, they will send signals to the plant telling it that these leaves are in the shade, that they aren't getting adequate light. As a response, some plants will simply stop giving sugar to the shade-covered leaves and let them wither and die. Instead, they'll invest this sugar in better positioned branches and better positioned leaves that have better access to sunlight. The plant isn't going to waste sugar investing it in a leaf that isn't even exposed to light. What's the point? It's analogous to building an oil drilling rig where there's no oil, or building a solar farm where there's no reliable sunlight, or building a wind farm where there's no reliable wind. The plant isn't going to misappropriate its resources like this. But other plants will take this shade signal and interpret it to mean that they're surrounded by other plants or natural light-blocking obstacles, and so they should grow taller to reach out above them. The stem will grow and rise upwards, hopefully bringing the shade-covered leaves out of the shade and up into the light. Now, on the other hand, when the phytochromes are in the FR conformation, this means that they've recently been exposed to red 660 nanometer light, and so they've recently changed their conformation to the P sub FR. When the phytochromes are in the FR conformation, they'll encourage the plant to germinate and to flower. When a seasonal plant gets signals of fewer hours of more and more red-tinged wavelengths of light caused by shorter days with a sun that rises lower in the sky, this will tell the plant that it's approaching the end of the summer, the end of a growing season, which will then go on to stimulate the processes that encourage sugar storage and seasonal dormancy. It takes a little bit more than just some red light to make a plant flower. Flowering is a very important part of the plant's life, involved in reproduction and the strategic timing of seed release. It's really important that flowering be timed with the day and with the seasons, so as to maximize the plant's chances at reproduction and having offspring spores or seeds that successfully germinate. Thus, the signal to start flowering is much more complex than simply being exposed to a particular wavelength of light. The length of time that they're exposed to light is also important. In the plants that live outside of the tropics, they tend to see seasonal changes in the amount of light. In higher latitudes, there are seasonal extremes of sunlight. In the winter, the sun hardly comes above the horizon, so it's really dark. And in the summer, the sun doesn't set, or if it does, it comes back above the horizon within just a few hours. Meanwhile, in the tropics, the sun comes up right overhead every day throughout the year, and so the amount of sunlight is relatively constant. The amount of light that a plant gets throughout the year depends on where it's located on the planet, and this amount of light is called the photoperiod. Plants will respond to various changes in their photoperiod that are appropriate for the season. For example, a plant that experiences an increase in the photoperiod, which is an increase in the amount of time that the plant is exposed to sunlight, well, the plant will sense this. It'll sense the increasing photoperiod, and it will deduce that winter is ending and summer is approaching, as the days, or the photoperiods, are getting longer. Conversely, a decrease in the photoperiod means that the days are getting shorter, and this indicates that summer is ending and winter is coming. Experiments have shown that the red, far red light switch is involved in triggering the flowering mechanism in plants. There are some plants called short-day plants. They bloom during the times in the growing season when the days are relatively short, like in the spring and the fall. These plants are pretty well accustomed to short days and long nights, and when these conditions are met, they will flower. Other plants are long-day plants, which thrive and bloom in the middle of the summer when the nights are short and the days are long. If you were to interrupt a long night with a brief but sustained flash of red light, say for about 60 seconds, you would actually flip the phytochrome protein switch and tell the plants that this time of year has short nights. As far as the plant can tell, the long night just got split up into two short nights, interrupted by a minute-long day of red light. This false signal will confuse the plants by making them think that it's a time of year with long days and short nights. The long day plants will then flower, and the short day plants won't. Now let's say that a few minutes after flashing the plants with red light, you decide to flash them with far red light. Well, this will flip the switch back to the way it was originally, and the plants will bloom normally. You can actually keep doing this, showing the plants multiple flashes of red and far red light throughout the course of a night, and in whatever position you leave the chemical switch, that will determine how the plants respond by flowering. But activation of the phytochrome, as I said, isn't the only thing involved in flowering. The plants have a circadian clock of sorts that involves cyclical increases and decreases in the concentration of various proteins and hormones as the day goes on. One of these proteins increases in concentration during the daytime, which stimulates a gene called constans, or CO, to produce an enzyme. This enzyme stimulates the expression of flowering hormones, but only when present in certain quantities. As the enzyme decays relatively quickly, its concentration in the cell doesn't rise very high, but when the phytochrome are activated by red light, they stabilize this CO enzyme and prevent it from decaying. So the CO enzyme is still getting produced, but it isn't breaking down, so it will accumulate in the plant's tissue and its concentration rises. In the course of a long day, a plant will thus accumulate a lot of this CO enzyme in its tissues. In a long-day plant, this will eventually reach a threshold where the CO concentration begins to encourage flowering hormones like florigen to initiate blooming. But in short-day plants, this high CO concentration inhibits flowering hormones and prevents blooming. The short-day plants are sensitive to shorter photoperiods, and so they only bloom when the time is right. As the seasons perpetually cycle through time, and the plant experiences day and night and day and night and day and night over and over again across its whole life, it'll be experiencing and sensing this cyclical, rhythmic expansion and contraction of the photoperiod in accordance with the seasons. As the plant ages and goes through its life cycle, it'll have various growth patterns and morphological expression and different responses that it makes to its environment. Different hormones are produced and expressed at different times in the plant's life. For example, the first bit of growth that a plant sees is from its apical meristems, where the shoot grows up out of the ground. This apical shoot has what's called apical dominance. This means that the lateral buds that can form branches will be suppressed by the dominance of the apical meristem growing upwards. But when this dominance effect fades as the plant ages, or if the dominance effect is shut down entirely by some herbivore coming by and eating the top of the plant and just fully consuming the apical meristem, well, in those circumstances, the apical dominance is lost, and the lateral buds begin to grow into branches, all with their own meristems at their tips. This dominance effect is created by the apical meristem producing auxin, which then gets shuttled throughout the rest of the plant's tissue. Now, you might be thinking that I've mentioned this chemical auxin a lot in this episode. It seems like it's the only hormone plants have. I mean, auxin helps roots find their way down. It helps shoots point towards the light. It helps regulate growth patterns, and it sustains apical dominance. Auxin is a really remarkable organic chemical because plants use it for this huge variety of purposes. It's almost like a fix-all hormone that plants can use for almost any circumstance. It's believed that auxin's most fundamental role is in giving the plant a sense of self-awareness. Now, I don't mean this in the way that we have self-awareness, but more like this. Because auxin is produced at the meristems, it exists in highest concentration at the tips of the stem and the branches. This auxin is then carried down through the cells from the apical meristems down to the roots. But as it goes down this pathway, as it moves across the plant's body, so much of the auxin will get diverted and used for other purposes by various cells and tissues on the way down, that by the time auxin actually gets to the tips of the roots, it exists in very low concentrations in the plant's tissue. There's another group of plant hormones called cytokinins, and these flow in the exact opposite direction to auxin. The cytokinins are mostly produced at the tips of roots, and they get carried upwards up through the plant to its shoots. But they're also expressed to some degree in places where cellular division is active, like inside seeds, or fruits, or buds, or the zones of division behind the meristems. These cytokinins, wherever they are in the plant, they play a critical role for a plant on a cellular level. They participate in the regulation of the cell cycle, which is the chemical process through which a cell divides and replicates itself in a stable, healthy way. Without these cytokinins regulating cellular replication, various organs of the plant, like fruits and flowers, can grow malformed. The seeds can be botched, and meristem growth can stall. The plant's body can chemically recognize the concentration of auxin in its cells, and the concentration of cytokinins, and as these chemicals exist in a gradient along the length of the plant, the specific cellular concentrations of each chemical can be used to identify where the cell is within the plant. And so, its body has the chemical self-awareness to know where certain tissues are, where certain types of growth should occur. Essentially, by identifying the concentrations of these chemicals and where these concentrations would exist along their gradients, individual cells can be accurately mapped to particular regions of the plant's body. There's other groups of plant hormones, like brassinosteroids, which help to stimulate plant growth, and ethylene, which is associated with senescence. In the case of brasinosteroids, these are kind of like plant growth hormones. Through means and mechanisms that are not entirely well understood, the brasinosteroids are able to alter gene expression to regulate the size of the plant cells during periods of growth. They can regulate the size of the plant, or at least the size of some of its tissues. The brasinosteroids stimulate stems to grow tall and branches to grow long. Now, ethylene is like the opposite of the brasinosteroids, because instead of growth, ethylene promotes ripening, and ultimately the rotting and the death of the plant tissue. Ethylene is the hormone responsible for telling fruits to begin ripening, which causes all manner of changes in the fruit to make it smell and taste and look more appetizing to animals. The purpose of fruit is to put the seeds in an edible container, so that animals like birds and primates will be attracted to that edible container and eat it. This way, the birds and primates and other fruit-eating animals can act as living shuttles for the seeds, transporting them away from their parent plant and helping the plant species spread out. When you see a really green bunch of bananas, they have very low levels of ethylene, but as they ripen, they become yellow. The husk gets softer, and the tissue becomes sweeter and smells better as starches are transformed into sugars. Now, I've mentioned dormancy a few times, like how some plants lose their leaves in the fall and go dormant over the winter. In the spring, dormancy typically ends, and the plant leaves dormancy and resumes its growth with the freshly watered soil and more sunlight. The plants use hormone signaling mechanisms to detect the oncoming seasonal change and make the appropriate physiological changes to accommodate it. In the fall, when the plant senses that the photoperiod is getting shorter and shorter and the temperature is dropping, a hormone called abscisic acid, or ABA, is released from the roots to inhibit growth. In the late summer, when the air gets cooler, rain is less common. The ground gets drier, and eventually it'll snow, and that snow will create a layer over the ground that prevents it from really receiving a whole lot more liquid water, at least until the snow melts. The point is, over the winter, the ground gets really dry. The root tissue can detect prolonged dryness and interpret this to mean that the long drought of the oncoming winter has arrived. This will stimulate the roots to release ABA, abscisic acid, which then travels up to the leaves and tells them to close their stomata, so as to conserve water. ABA is a dominant signal that overrides the signal from blue light to open the stomata. In the seed, ABA interacts with another group of plant hormones called gibberellins to influence the length of dormancy and the timing of germination. The gibberellins have an internal biochemical role that's the opposite of abscisic acid. Where ABA inhibits growth, gibberellins will promote growth. So many plant seeds have enough sensory processing capacity that they can tell when conditions outside the seed are prime for germination, or if things aren't quite good enough. For example, the seeds can tell if the soil around them is moist enough or warm enough. Signals that tell the seed that it's not a good time to germinate will instead encourage the expression of ABA, which will bind with specific receptors on the membranes of the cells and initiate a signal transduction cascade within the cells that activates specific transcription factors that will then promote the expression of specific genes. And in the case of abscisic acid, these genes that ultimately get activated at the end of this biochemical pathway, these genes will inhibit growth and prevent germination. But when conditions are more favorable, when the seed can tell that the soil around it is fertile and the time of year to grow is optimal, then all of these environmental signals will encourage the production of a gibberellin called gibberellic acid, or GA. When GA is released into the cells of the seed, it can activate genes that express an enzyme called alpha amylase. ABA, in contrast, suppresses alpha amylase expression. And this is a really important difference, because alpha amylase is an enzyme that breaks down starch, and turns it into bioavailable sugars. When there's a high concentration of alpha amylase in the seed, it diffuses into the endosperm and begins to break down all of the starch that's been stored there. This breakdown of the starch will release a lot of sugar into the cells, and this wakes everything back up out of dormancy and initiates germination. The sugar is used to promote the plant's early germination and initial growth, like the protruding taproot searching for soil, and the cotyledons unfurling into leaves. Inside the cell, the concentrations of ABA and GA are in perpetual flux. The concentration of these hormones in the plant cells is really important, because the concentration of the hormone is directly associated with whatever the hormone does. In this case, the hormone initiates signal transduction cascades involving signaling proteins and enzymes that help read and express DNA. Now, as the winter season comes to an end and the spring appears, environmental conditions will cause less abscisic acid to be expressed. Its concentration in the tissue will slowly fall, and its capacity to suppress the expression of alpha amylase is slowly reduced. Simultaneously, these same conditions will cause more gibberellic acid to be expressed, and its concentration in the seed tissue will rise, which further stimulates the expression of alpha amylase. Gradually, over a brief period of time, the concentration of gibberellic acid and alpha amylase will more and more overshadow the concentration of the abscisic acid and its inhibitory effects, and eventually sugar will begin to diffuse through the seed's tissues. Germination slowly ramps up as sugar accumulates and the cells in the seed reawaken with life and begin using all that sugar to power their growth and replication. And all of this is regulated and propagated by the seed's sensory inputs, by its capacity to sense the environmental conditions around it. One of the last things that I want to talk about today are the signals that plants use to detect and respond to infection, and how they respond to the mechanical damage caused by an herbivore coming by and chewing them up. When a plant gets infected with a bacterial or a viral pathogen, its first line of defense is to close its stomata in the infected area to prevent further infection. This is the first step in the hypersensitive response, which is the plant's defense against these kinds of microbial invaders. They don't have an immune system per se, but they do have the hypersensitive response. So after the stomata are closed, the plant produces toxins in the local area that will fight off the pathogen in a way that's not dissimilar from the animal immune system. This even involves the chemical reinforcement of nearby cell walls, so as to create a literal cellular barrier, a blockade, to mechanically contain the pathogenic particles. Even more radically, the plants will sometimes engage in a scorched earth policy by killing its infected cells to deny their metabolic products to the pathogen. As this hypersensitive response is going on, the plant is also sending out warning signals to nearby cells, which initiates a slower but more widespread response called the SAR, or the Systemic Acquired Resistance. This primes other cells in the plant to release defensive chemicals at the first sign of pathogen infection. One of the most common, but energetically and nutritiously expensive means that plants have to repel a potential herbivore is by producing a substance that tastes or smells really bad, something that stings or is uncomfortable to be around. For example, the oils that are produced by herbs like lemon, sage, and peppermint will repel insects, which try to come and eat their leaves. These oils create intense odors and tastes that will just fry the little insect sensory buds and scare them off. Some pine trees will outwardly secrete a chemical called pinene, which will poison any bark beetles that might have been burrowing around in the tree bark. A lot of plants will express proteins called Protease Inhibitors, and as the name suggests, these proteins will disable the enzymes that break down proteins as part of an animal's digestion. As a result, if an herbivore eats too much of a plant, it will ingest too many of these protease inhibitors, and the animal will experience difficulty breaking down the proteins in its own cells, which can lead to illness. The animal is thus discouraged from eating that plant, or at least from eating too much of it in one sitting. If the herbivorous animal takes a bite into a plant and tastes a high concentration of these protease inhibitors, then the animal will generally be disgusted by the taste, and it'll move on. It won't want to eat the plant. Now, what's really crazy is that if the plant tissue, like leaves, is getting physically destroyed by the mechanical chewing action of an herbivore, that pulpy, wounded tissue is able to release a protein hormone called cysteamine. This is moved through the phloem, where it comes into contact with numerous plant cells and activates receptors on their cell membranes that initiates the synthesis of jasmonic acid. This jasmonic acid will then go on to stimulate the expression of various genes, including the genes that produce the protease inhibitors. So, by eating some of its leaves, an herbivorous animal can cause the plant to release a chemical that will eventually make its leaves taste bitter and terrible, so after just a few bites, the animal is encouraged to move on to a different plant. The benefit to the plant that was being chewed on is that it doesn't have extensive tissue damage. The plant isn't so wounded that it's going to die. It can still live another day and photosynthesize and reproduce and do all the things that plants do. But if you thought that was pretty wild, well, it gets a little crazier. Some plants will even release an airborne signaling chemical that can tell other plants, "Hey, I'm being eaten over here. There's an herbivore here that's eating me." And this emergency signal will cause all of the other plants in the nearby area that have detected that signal to produce more protease inhibitors and various defensive chemicals like toxins and embittering compounds. Some plants will even release chemicals that attract predatory insects like wasps, and these predatory insects will come in and hunt down whatever kind of herbivore is eating the plant tissue. There's plants that do this to recruit wasps to come and kill caterpillars. It's a kind of interspecies defense mechanism. Alright, before I end the episode, I want to mention one more defensive measure that plants have. Sometimes, plants will produce a chemical that has a physiological and neurological effect on the animals that consume it. The idea is that this effect is uncomfortable, and it will discourage animals from eating those plants, but in some cases this can backfire. Caffeine, cocaine, opium, and nicotine are all plant-based chemical compounds that are intended to scare the herbivore by disturbing its sobriety with a mind-altering chemical. The plant is basically sending the message, "Don't eat me, or I'll mess with your perception of reality." For most animals, being stoned or incapacitated like this can be dangerous, because they can't readily defend themselves or maintain sufficient awareness of their surroundings. Humans, however, will cultivate these plants and consume them specifically to experience the mind-altering effects that they give. This includes everything from the coffee you drink in the morning to wake you up and make you a little more productive at work, the joint that you smoke in the afternoon to relax after work, and the cigarette you have on your work break to the opium products produced from Afghanistan's poppy fields and the cocaine produced from the jungles of Colombia. All of these drugs are plant products that the plant has evolved as a way of scaring off or repelling the herbivores who wanted to come and eat them. So with that said, I think I've reached the safe limits of digression away from plant sensoria, while still keeping things at least tangentially related. If you like this episode, then hit the like button, and if you want to catch all of my content right when it's posted, then be sure to go to my YouTube channel and hit the subscribe button. If you're listening to this episode on iTunes, then do me a huge favor and give the podcast a favorable rating, and leave a positive review. All this stuff really helps the podcast. It helps get the word out there, and for that, you have my gratitude. And as always, thanks for listening.